Is it actually possible to use CRISPR as a gene editing tool for higher intelligence? The short answer is maybe in the future, but it is heavily dependent on future advances in synthetic biology studies and gene manipulation methods, which have to be more precise than CRISPR, base editing, prime editing, or any other direct engineering methods in vivo. So, there are a myriad of limitations and possible tasks we gotta deal with first in order to ever consider those types of therapies, which might be utilized later if we ever want to actualize those types of advances in therapeutic approach. The first one, and most likely the most important, is the approach. The intelligence is highly polygenic trait, meaning that as it is for, say, sickle cell disease, where mutation causing this type of genetic disorder is caused by the single nucleotide change, or for the intelligence, there are hundreds, if not thousands, of genes, which by themselves contribute for special types of protein synthesis, enzymatic activities, which alone might not do much, but overall, if all these thousands of genes were put together, would be fully responsible for many cognitive activities and intelligence determining factors. If we could ever achieve finding all these genes, determining their coding sequences without introns, which are non-coding segments of eukaryotic DNA, we could, in theory, overexpress the gene synthesis, and perhaps this overexpression would lead to enhanced neural activities. The second factor is the editing method. Yes, CRISPR is revolutionary, and yes, without CRISPR, further advances would be unimaginable. But as it is right now, the whole method is still far from perfect. We cannot effectively use CRISPR-Cas9 protein for cleaving longer base pair genes, and many intelligence factors are composed of thousands, if not tens of thousands of base pairs. So CRISPR, as it is right now, couldn't possibly be used facing sequences this large. The third limitation we absolutely gotta overcome is the carrier mechanism. In human applications, researchers mostly use AAV, meaning adeno-associated viral vectors. These are truly the revolutionary, tiny, cute viral particles, but sadly for us, they can only carry the limited number of hereditary luggage. On average, the number is 4 to 5 kilobase pairs, which for our taste is too goddamn small. In theory, we could use retroviruses, but the odds of allergic reaction is quadrupled, and simultaneously, most retroviruses are RNA type, not DNA. So, in addition, they have to reverse transcribe their single-stranded RNA into the double-stranded DNA, which further complicates the things. To sum up, if we manage to find way effective carrier virus, more versatile Cas protein, and determine all the genes, all the coding exons, and all the start and termination codons, synthesize the synthetic proteins, study them in animal models, and further stabilize those genes using crossbreeding, we could accumulate the knowledge large enough to overcome every obstacle and get a step closer to attaining Homo Deus.